After a blood specimen has been diluted as directed by manufacturer instructions and the vial has been mixed, a small amount of specimen is loaded into the hemacytometer chamber for counting. Materials needed for this process include hemacytometer with cover glass, diluted specimen, alcohol and lint-free tissues, timer, moist chamber, and a microscope with 10x and 40x objectives. The chamber is composed of the hemacytometer and cover glass. This cover glass is specific for the chamber and is optically flat to ensure a consistent depth over the entire counting chamber. The center section of the hemacytometer has three components: the platforms, the moat, and the bridges. There are two platforms, one on each side of the chamber. As you look closely at the platforms, you should be able to see a grid on each side. There is a V-shaped groove on each side that is used to help load the specimen into the chamber. Around each platform is a moat which separates each platform from the other. Beside the moat on each side are the bridges. These raised sides provide support for the cover glass, and the distance between the platforms and the cover glass is 0.1 millimeter. This space will be filled with a fluid on which the cell count will be performed. Accurate cell counts depend on the quality of the equipment used as well as using proper technique. Each platform is etched with a large grid measuring 3 millimeters on each side. This large grid is subdivided into 9 1 millimeter squares. Each square is subdivided into various additional squares for additional utility. The first step to correctly loading the hemacytometer is to ensure that it is immaculately clean. Any debris left can negatively impact the optical quality of the chamber and cause incorrect results to be obtained. The chamber is cleaned using isopropyl alcohol and lint-free tissue. The cover glass should always be handled by its edges. To begin loading the chamber, the cover glass is placed on the hemacytometer so both sides of the chamber are covered equally. Next, the diluted specimen is mixed gently but thoroughly, and the first few drops of liquid are expressed into a tissue. While maintaining pressure on the specimen vial, the next drop is applied to the V-shaped groove at the junction of the cover glass and chamber. Pressure is continuously applied to the vial and the pipette is held at a 45-degree angle. The slight pressure on the vial will work with the capillary action of the glass to properly fill the chamber. Watch carefully and remove the pipette from the chamber when it is completely filled but not overfilled. If fluid flows into the moat, the chamber is overfilled and the chamber will have to be cleaned and the specimen reloaded. If it will be several minutes before counting can occur, the chamber should be placed in a moist chamber. This can be created by placing a damp filter paper in an empty Petri plate raised on pieces of wooden applicator sticks. The hemacytometer is allowed to sit for 1 to 2 minutes to allow the cells to settle for counting. The chamber is carefully placed on a microscope for counting.